Our next speaker is Serena Martinez. Terina is Executive Director of two rare disease consortia for C-Path, the Duchenne Consortium and the CPTA, or Consortium for Rare Ataxias. Terina has a PhD in integrative biology from the University of Texas. She completed her postdoc in Pittsburgh. Before joining C-Path, she was working for the Michael J. Fox Foundation, leading preclinical, the development of programs for preclinical tools surrounding the topic of inflammation in neurological disorders. And she also worked for a biotech called Taconic Bioscience before joining C-Path. So welcome, Terina, to the stage.
Thank you, Alex, for the introduction, and I'm so pleased to be with everyone today and in person. That's, another benefit to bringing us all together. So as Alex mentioned, I am going to speak to you today about some efforts that we are working on in our Duchenne Regulatory Science Consortium and how we are leveraging data from this rare disease to have some solutions to help improve clinical trial enrollment. I have no disclosures. Duchenne is a rare disease, neuromuscular disease, that is characterized by monogenic mutation in the gene dystrophin, which is a crucial component of muscle, and it's necessary to build strength and recover from mechanical stress and injury. So without dystrophin, there's damage to the muscle, and that leads to dysfunction that gets worse over the lifetime. This is a disease that's rare and affects approximately 1 in 5,000 males because it's X-linked, and it is very early onset, usually noticed in children because they aren't making their motor milestones. The challenge of the disease, of course, is the nature that it's rare. It is insidious. It is affecting children over their lifetime and is progressive and leading to significant disability and death. The characteristics of the disease through the progression are shown on this chart, where you can see that in the early years, the individuals with the disease are still ambulatory, but they reach a stage where they transition from ambulatory to non-ambulatory. And then once this occurs, they then require ventilation support, wheelchair use, and continued disability. DMD is very progressive and fatal. There are limited therapies. The current standard of care is glucocorticoid therapy. So the steroids do help reduce the inflammation in the muscle and can prolong lifespan to a certain extent, but quality of life is still dramatically affected. There has been some recent progress in drug development for DMD. This has yielded a few regulatory approved treatments. So one of these treatments is a steroid that was just shown to be more effective than prednisone. So deflazacort is more effective than prednisone for Duchenne. And then a few other drugs that are focused on exon skipping. But this means that they're only applicable to a minority of the population that has Duchenne. So much more work still needs to be done. The failure rate for DMD clinical trials is high. Actually, Sergey set me up for this because he showed that there's been something like 54 clinical trials for Duchenne, but only a few approved drugs. So this failure rate is very high, and there's a need for novel and diverse strategies for drug development. In this schematic, we're really looking at the complexity of Duchenne. So if you consider the disease over its continuum, initially, there is an opportunity to intervene early before the muscles get fully degenerated. And then over time, the disease progression is characterized by inflammation, fibrosis, integration of fat into the muscle. And so the approaches that would be taken would probably need to be different across the spectrum. So if you're intervening early, the approach is going to be different than if you're intervening in an individual who's had the disease much longer. There is complexity to this, though. Like with so many other diseases that are rare, in addition to the challenges of the rareness, there are disease-specific components to Duchenne that provide additional complexity. Dystrophin is one of the largest genes in the human genome. It's absolutely massive. It is 2.4 megabases, and the translated protein is 427 kilodalton. This means that it exceeds the packaging capacity of most viral vectors for gene therapy. So an approach to make a microdystrophin is used instead. And because it's a monogenic disease, it is a treatment that is amenable to potentially gene therapy or exon skipping strategies. Because skeletal muscle is post-mitotic and maintained by repair and regeneration by a population of stem cell-like satellite cells, cell replacement therapy is also an option. But targeting muscle is very difficult to do. It's a large organ, it's post-mitotic. I have mentioned the molecular challenges of designing drugs for Duchenne. And all of this is layered into the background of the complexities of the population of individuals who have the disease because of the rareness, the pediatric group, et cetera. So this really highlights the many challenges for DMD drug trials. I mentioned the fact that it's a rare disease in a pediatric population, the complexities of the target itself, the complexities of disease progression. There's actually an initial phase when the boys are growing that they actually can look like they're improving in some of their measures of functional outcome just because of growth, but eventually the disease progression catches up and they have worse function from there. There's also a general lack of good clinical endpoints that span the entire disease continuum. As I mentioned, because of the way the disease progresses, there isn't one single scale or measure that can really be assessed at all stages. And there's this variability of disease progression as well. So even though it's a monogenic disease, because the gene is so large, the number of mutations and where the gene and the protein are disrupted can lead to large heterogeneity in how the individuals progress through the disease stages. So that's why the Critical Path Institute developed the Duchenne Regulatory Science Consortium. This is a global collaboration formed by C-Path and the parent Project Muscular Dystrophy group, which is a patient advocacy group and research foundation, to convene pharmaceutical industry members, academic and clinical researchers, nonprofit foundations, and patient advocacy groups to develop tools to accelerate therapeutic development for Duchenne Muscular Dystrophy, which I'll abbreviate DMD. D-RSC, which is the Duchenne Regulatory Science Consortia members, include 12 biopharmaceutical groups, 2 patient advocacy groups, 15 academic and clinical centers, and regulatory and government advisors. Our goal is to combine and aggregate the data so that we can enhance the utility of it and use it to drive data-driven decisions to make solutions to improve therapeutic advancement for Duchenne. The 4 main buckets for the efforts that we have within D-RSC focus on data sharing and analysis, biomarkers, complex trial design, and tools to optimize patient selection for clinical trials. I'm going to really highlight 2 components for this presentation today, focusing on the data and how we're using that data for a clinical trial simulation tool. This is all built, of course, on the data, and that's really the key to everything that we're working on in D-RSC. The D-RSC integrated database has been built over the last 6 years. It includes 13 clinical trials, 5 natural history studies, 3 clinical studies, and 1 patient registry. This is a really rich data set that allows us to combine the opportunity to evaluate natural history data and clinical trial data. Unfortunately, because so many of the trials fail, we're looking at placebo arm data because there's not active drug data available because so many of the trials fail. But really, this data set is the key and anchor to everything that we're doing in D-RSC. And I certainly want to credit and thank the data contributors and the individuals with DMD and their families who participated in the studies and consented to sharing the data. So I now want to shift gears and show you how we're using this data in order to drive solutions for clinical trials in DMD. This is the who, what, why, how slide. So what we're looking at is a quantitative solution of disease progression models to allow in silico evaluation of relevant parameters to optimize trial design. The how is by leveraging that rich database that I just mentioned, the database with patient-level data for individuals with Duchenne muscular dystrophy obtained from clinical trials and natural history studies in order to train and validate computer models to simulate parameters relevant to clinical trial design strategy. The who, not the band from the UK, but the who are the people who would be utilizing this tool. We're envisioning clinical trialists, researchers, regulators, and advocacy groups all to be able to have benefit from this tool, which will be publicly available. The why is, of course, because of the urgency needed within the Duchenne community and to improve trials for this disease. We really do not have similar quantitative solutions. There's no other approach that can be improved upon. This would be the first one. And there is the urgent need to optimize the clinical trials for this disease so that fewer participants need to be enrolled and so that clearer outcomes can be achieved. A clinical trial simulation tool, which I'll now abbreviate as CTS in the following slides, for DMD could truly help deliver the right drug at the right time to the right patient. So this again is all built on the data. The input of this clinical trial simulation tool, or CTS, is the clinical trial data and natural history data from these observational studies. It's then kind of mined from that rich database that I just mentioned. The data is then mined and interrogated to assess several components that are of interest for a clinical trial simulation tool, which are shown in these 3 columns. One is looking at disease progression features. This could cover demographics, clinical assessments, and biomarkers. Another column looks at clinical trial components, such as the placebo effect, trial design, and dropout. So when the subjects have to drop out from participating in the trial. And then also candidate drug effects. This could be looking at dose response, disease modification, or symptomatic features. All of this is kind of within this bigger system of the model training and validation, and the output of everything is the clinical trial simulation tool. The goal of this clinical trial simulation tool and the strategy behind it is to develop a simulation platform that will enable in silico evaluation and refinement of clinical trial design for use in the development of therapies for the treatment of DMD. So it's an iterative and machine learning process where the trial information and the data from the database that includes all of that information that I mentioned on the previous slide, the demographics, the outcome measures, et cetera, is input into quantitative models that then simulate the results. So we're simulating disease trajectories and progression from the real data of the individuals who were in the trials or in the natural history studies and simulating what that looks like over time. And then that is analyzed statistically so that we can continue to fit and train and improve the model so that at the end you have a model that is representative of the global Duchenne community and can be used by sponsors to help improve clinical trials. And again, it's important to be able to look at this complex trajectory. As I mentioned earlier, it's biphasic, starting with the initial improvement in function and then the decrease in function, and that complexity of heterogeneity, that complexity of individual response and progression through the disease. We need to be able to capture that. And by being able to look at many individuals aggregated through the database and then training the model, we're able to simulate even more. So the tool is built, again, on the data. We have 2 versions of the tool because they are being submitted to both the EMA and the FDA. This is the slide that outlines how the different data sets are integrated into the 2 versions of the tool. Again, we have many data sets in the DMD database, but we needed to find the data sets that had the outcome measures that we were looking at. Not every study is going to have every outcome, but we are able to have several studies or trial data sets that overlap the different outcomes that we're looking at. So this summarizes how the tools are built for the EMA model. There's a certain subset of the data that was used for training, and then a different subset that was used for validation. For the FDA version of the model, it uses a standard 80-20 split. So the entire data is taken in totality. 80% is randomly selected to train, 20% is used for validation. We are submitting this to the EMA through the Qualification of Novel Methodologies pathway. And this was work that was done by the quantitative medicine team at C-Path and with collaborators at the University of Florida. And for the FDA model, we're submitting this through the Fit for Purpose Initiative for Drug Development Tools. And this modeling work was done with collaborators at the University of Ljubljana and by the C-Path quantitative medicine team. So the endpoints, as I mentioned, we have these multiple endpoints that we're looking at because we want to model disease progression. We want to first assess these endpoints at baseline and then look at them longitudinally over time so that we can understand on a population-wide basis what the variability of the disease progression is, so that we can understand the variability in the clinical endpoints, understand subpopulations. Certain individuals with certain types of mutations are fast progressors and some are slower progressors. We need to understand that within the broader continuum of the disease. And we also want to understand the effects of trial factors such as dropout, placebo, et cetera. These longitudinal models incorporate relevant sources of variability such as baseline severity, age, steroid use, genetic mutation, and also study type, whether it's the clinical trial or the natural history observational studies. These models incorporate data from commonly used functional assessments that have really been mainstay within the Duchenne community for several years and are widely used in the clinical trials. This includes the North Star Ambulatory Assessment, which is a total score of a 17-item rating scale that measures functional motor abilities in ambulant individuals with DMD. We also have 3 velocity tests. So these test the time that it takes an individual to rise from supine to stand, to climb 4 stairs, and to walk 10 meters. We also included the 30-foot walk because it's really an equivalent distance, and that allowed us to bring in more data for this measurement. And we have one other additional functional measure that we're looking at, which is forced vital capacity. This is a pulmonary assessment. Because Duchenne affects muscle, it affects skeletal muscle in addition to diaphragm and also cardiac muscle, which is why the need for nocturnal ventilation and treatment for cardiomyopathy are also required, especially in advanced disease. So as I mentioned earlier, there's no single outcome that measures the full spectrum of Duchenne, but by incorporating these multiple endpoints, we're able to approximate the full continuum of the disease. And forced vital capacity is one that is very important in later stages after ambulation has progressed to non-ambulatory. So these models simulate the effects of these known covariates on progression for each endpoint across the disease spectrum. This clinical trial simulation tool supports simulation-based estimates required for power calculations. It allows users to implement hypothesized drug effect parameters and in an endpoint-specific manner, and it features a user-friendly graphical user interface, or GUI. So the next couple of images are going to be screenshots and representations of what the simulator does. So this is how a user would interact with the simulation tool. They would start by choosing an endpoint. In this case, we're selecting the North Star Ambulatory Assessment. You'd then be able to look at other features that are important to the clinical trial, such as the opportunity to input your baseline parameters, and then also to look at how you can incorporate the different features, the estimations for sample size using sliders to look at trial duration and other features. And then you can use that to simulate what, based again on that data that's been trained and validated, simulate what your parameters that you've inputted into the tool will help you understand what your trial would look like. And this is an example of that. This is a representative scenario of simulation for hypothetical drug effect by time and study. Again, looking at NSAA and being able to look at the size of the population, the duration of the clinical trial, and in order to extrapolate what the drug effect could be. Certainly, these models are not intended to replace functional assessments in clinical trials for the assessment of efficacy. But we do posit that because of the richness of the data that built this tool, it's representative of the global Duchenne population, and these models do provide a rational basis for decision-making on quantifying how effective trial design is to reach the objectives. It is important to seek regulatory approval for tools like this, not just, there are several reasons. Some of the main ones are that it increases the confidence in the rigor of the tool. It enables familiarity of the tool components and its utility across stakeholders. And one of the things that I think is really key to this process also is that regulatory approval of the tool has a strong effect on stimulating additional data sharing. It's a really tangible way, an accessible way for people to see the impact of sharing their rare disease data. And for, as we spoke earlier, as the panel discussed, the reticence sometimes of people sharing the data, whether they're industry sponsors with clinical data or others, being able to pull it together and build a tool like this that can then be used publicly is a very meaningful way to increase the data sharing. So the status update for the tool, because it is matriculating through both of those regulatory pathways, the fit for purpose of the FDA and qualification of novel methodologies at EMA. Today, we submitted the briefing package to the FDA for this tool. So this is moving forward to that next step. And 2 weeks ago yesterday, we had our discussion meeting with the EMA for that submission. So it's really great to see this resource intensive, deep richness of data, and in several years of developing this clinical trial simulation tool get close to the milestones and deliverables of the finish line. So once these tools are qualified, they will be made freely available to the global DMD community, and we will continue to evolve and optimize them so that we can make them even better. As more trials are conducted, as active arm data becomes available, we'll be able to do even more with the foundation that we're building through these tools. And just in the last summary, I've given you a glimpse, I hope, of the clinical trial simulation tool and how we're leveraging data within the Duchenne Regulatory Science Consortium. And I just want to give a little bit of a forecast for some of the other projects that we're using the data for within the program. We're continuing to expand our database. Currently, we're focusing on cardiac data and active arm clinical trial data. And we're also leveraging our database to explore and model specific analytic questions, some that we're looking to move forward into our outcome measures applicable to early disease states and the transition state from ambulatory to non-ambulatory. As I mentioned, we're really interested in the cardiac data. One of the key challenges for cardiomyopathy in Duchenne is that even the therapies that are pursuing the gene therapy modalities have poor tropism for cardiac muscle. So there could be scenarios where we have improved function in skeletal muscle, but are not moving the needle in cardiac function. So it would be a difficult scenario to give an individual more capacity to exercise, but then their heart can't handle it. So there's much to be done here. And we are starting off early with the data because it needs to be standardized. It's the Wild West right now for cardiac data in Duchenne. We really need to standardize it, harmonize it, and understand how to translate the data into interoperable formats. And then we are finishing our regulatory submission of another clinical trial simulation tool. This one's looking at disease progression models for functional tests and also muscle fat fraction, because you can quantify and measure the amount of integration of the fat fraction in the muscle and relate that to progression and loss of function. This is in partnership with Imaging DMD. But these are the data projects that the Duchenne Regulatory Science Consortium is working on. Clearly, this is what keeps me up at night, is trying to understand how to really accelerate therapy for this devastating disease. Thank you for your attention.